Hello and welcome everybody to my channel Omni Bio Entrance, and today I will be dealing with Telangana State CPGET MSc Biotechnology Entrance Exam question paper conducted in the year 2023. This is part 2. Part 1 I have already uploaded. You can refer to my channel playlist for the same. If you are new to my channel, please do like, share and subscribe. Coming to the first question. To promote attachment and spreading of anchorage-dependent animal cells, the surface of the culture vessel need to be coated with. So, for that you need to coat the culture vessel with collagen. Answer is option 2. The next one. Which one of the following is not a part of the human non-specific defense system? So, it is antibody, because antibody is a part of specific immune system. Then, which one of the following amino acid is catalyzed by activated macrophages to produce reactive nitrogen species? It is arginine. Answer is option 1. All these are direct questions. Then identification of blood groups involved. So it involves the principle of agglutination. Answer is option 4. Then siRNAs interfere at. So siRNA, it is interfering at the post-transcriptional level. Then which of the following is a 39 residue hormone of the anterior pituitary gland? It is corticotropin. Answer is option 3. Most abundant RNA in the cell. It is rRNA. Answer is option 1. A high content of triglycerides are found in. So they are found in chylomicrons. Answer is option 4. Which of the following is a phospholipid? So out of these, phosphatidylcholine is a phospholipid. Answer is option 1. Then which one among them is the example of competitive inhibition of an enzyme? So succinate dehydrogenase by malonic acid. It is an example of competitive inhibition of the enzyme. In competitive inhibition, the inhibitor competes with the substrate for the binding to the active site of the enzyme. So the answer here for example of competitive inhibition of an enzyme is succinate dehydrogenase by malonic acid. Then cystic fibrosis. Cystic fibrosis, it is an autosomal recessive disorder. Answer is option 3. The phenomenon known as chromosomal separation can occasionally separate alleles of different genes on the same chromosome. That is, the phenomenon is called as crossing over. Which of the following statement is not true? A DNA is wider and longer than B DNA. A DNA is wider than B DNA, but it is shorter than B DNA and not longer. So first statement itself is not true. Then next one. The first human chromosome to be fully sequenced as part of the Human Genome Project is. So it is chromosome 22. Answer is option 4. Then glycolysis can occur in. So it can occur in both aerobic and anaerobic cells. In aerobic condition, pyruvate enters citric acid cycle and it will undergo oxidative phosphorylation. Then in anaerobic conditions, pyruvate converts to lactate. Then next one. In which of the following phase secondary metabolites are produced during growth? So it is the phase. Answer is option 3. The next one. Which of the following carrier gases are generated at on-site in GC? So nitrogen and hydrogen. Oxygen is not used as a carrier gas because it is highly reactive, and the possibility of unwanted reactions will be there when you use oxygen. So oxygen is not used as a carrier. Then which enzyme is responsible for the initial step of beta oxidation? So first step of beta oxidation is of the fatty acid by acyl-CoA dehydrogenase. So answer is option 1. Which of the following is an example of cellular component of the innate immune system? So cellular components, they include the phagocytic cells, epithelial cells, endothelial cells, natural killer cells, and innate lymphoid cells and platelets. So the cellular components out of these options is natural killer cell. Answer is option 3. Then which microscopy technique utilizes a beam of electrons to create an image of the specimen? So the beam of electrons is used in the electron microscopy, and there are two types, that is scanning electron microscope and transmission electron microscope. So 3 and 4 both answers are correct. Then next one, which type of media is used to distinguish between different microorganisms based on their metabolic properties or appearance on the media? So it is the differential media that is used. Answer is option 2. Then next one, which technique allows for the detection and quantification of gene expression levels in a sample? So it is reverse transcription polymerase chain reaction or RT-PCR. Answer is option 1. Then which of the following is an example of genetic engineering technique used to modify the DNA sequence of an organism? So polymerase chain reaction, no. Western blotting is used to separate and identify proteins. DNA microarray analysis is to measure the expression level of a large number of genes simultaneously. So that is also not used. CRISPR-Cas9 gene editing, that is used to modify the DNA sequence of an organism. So answer option 4. Then which of the following factors is required for the initiation of translation in eukaryotes? Shine-Dalgarno sequence, no. Cap binding complex, EIF4F. So that is required for the initiation of translation in eukaryotes. Answer is option 2. Then products of glucose oxidation essential for oxidative phosphorylation are. So the end products of Krebs cycle of glucose oxidation are carbon dioxide, NADH, FADH2, ATP and water. Now these end products, they serve as reactants for oxidative phosphorylation. So the reactants of oxidative phosphorylation are NADH, FADH2, oxygen, ADP and PI. So the answer here is option 4, that is NADH and FADH2. Which of the following enzyme is responsible for the regulation of biological nitrogen fixation? It is dinitrogenase reductase. Answer is option 1. Then which observation was made by Avery, MacLeod and McCarthy? So they are involved, these three scientists, they are involved in confirmation of transformation experiment carried out by Griffith. So they have found that DNA is the transforming principle. So DNA is a duplex molecule. No, DNA can be taken up from the medium and can be used for transforming. So Option 2 is the right answer. DNA can denature at higher temperatures and DNA is more stable than RNA. These two options are not related to transformation principle. So only option 2 is related. So answer is option 2. P1 cloning vector is an example of. So it is an example of a bacteriophage. Cosmid vectors are used for. So cosmid vectors, they are used for cloning large fragments of DNA. Polysomes are aggregates of ribosomes. No. Aggregates of lysosomes. No. mRNA molecule to which many ribosomes are attached. That is polysome. Answer is option 3. What are asters made of? So aster is a cellular structure and it is shaped like a star and found around each centrosome during mitosis in an animal cell. They are not formed during mitosis in plants. So the asters, they are made of microtubules. Answer is option 4. Then next one. In severe combined immune disorder, the patient is deficient in. So the patient is deficient in both B cells and T cells. So the answer is option 3, that is both A and B. So the patient here in SCID is deficient in T cell and B cell function. So the answer is option 3. The next one. Which one of the following is a cell-mediated hypersensitivity reaction? So it is type 4. Then the last question. If the value of chi-square calculated is higher than the value of chi-square critical, what step a statistician should take? Now if the chi-square value does not exceed the critical value, then the null hypothesis will be accepted. Now if it exceeds the critical value, the null hypothesis will be rejected. So the answer here is option 1, that is reject null hypothesis. Because in the question they have asked when chi-square value calculated is higher than the chi-square critical value. So reject the null hypothesis. So thank you for watching my channel. Please do like, share and subscribe. I have uploaded part 1 also. You can refer to my channel playlist for the same.